Hello, my name is Ladislav Bumba, and I'm working as a postdoc at the Institute of Microbiology in Prague, in the Czech Republic. So in our lab, we are interested in studying host-pathogen interactions, and besides all the molecular methods like we are using, we also using the SPR machine, which serve us for studying the interactions between the toxins and some effectors from bacteria and their interaction with the animal receptors on the cell surface. I would like to invite you to SPR ProteOn XPR36 webinar, where I will summarize the advantages of the SPR machine, and I will share my real measurements and experiences with this instrument to a broader scientific community.
Okay, thank you. Thank you for your introduction. And I will present my recent data. As you mentioned, the ProteOn XPR36 machine was bought by our institute 2 years ago, and I'm the person who is responsible for managing this instrument and for measurement. So surface plasmon resonance is an optical biosensor. Okay, so surface plasmon resonance is an optical biosensor which doesn't require neither radiochemical or non-fluorescent labels to provide real-time data on the affinity, specificity, and the kinetics of protein interactions. And it can detect picomolar levels of proteins and other molecules as small as few hundred daltons. SPR occurs when light interacts with a metal film placed at the interface between two media with different refractive indices such as glass and water. SPR biosensors respond in real time to changes in the refractive index at the interface resulting from the binding and subsequent separation of two molecules, one which is bound to the sensor surface. As the molecule in solution, which is analyte, flowing over the surface, bind to immobilized molecule, which is ligand, and the refractive index near the sensor surfaces increases, leading to a shift in the SPR angle. When the complex on the sensor surface dissociates, the SPR angle shifts back. So these changes in the refractive index are expressed in relative units or response units and are proportional to mass changes at the surface of the sensor chip. So on the slides you can see you can see the schematic representation of the surface plasmon resonance instrument. There is a light source which is shining into the glass prism, and the light is reflected by the prism, and in the detector is detected the refractive index of the instrument. So on your screen you can see the association curve and dissociation curve. So in the next slide there is a picture of the Bio-Rad ProteOn XPR36 system multiplex SPR, which is on the there's a computer which is connected to the SPR machine. There is a chip, and there is a robotic machine for the auto sampling, and there are some buffer storage. In comparison, to standard SPR machines, which can be seen on the left side, where in standard approach, the determination of kinetic ray constant have been run sequentially using two flow cells where one is a reference and one is for binding interactions. The reference cell didn't contain any ligand and served to measure non-specific binding of the analyte to the surface of the chip, bulk effects, and signal drifts. But the disadvantage of this is that the single concentration of analyte is flowing over the ligand of interest bound to the surface in the sample flow cells, and the corresponding response data are collected. The reference cell signal is subtracted from the signal in the sample flow cells. So this is a very time-consuming procedure, and this can be in comparison to ProteOn XPR36 system. So there is a 6-to-6 interaction array, which is seen on the right side of the slides. There is a glass prism with the 6-to-6 arrays, which makes the 36 interaction points. But of course, there are also interspots, which is between the interaction spots, and there are 42 vertical and 42 horizontal interspots. And these interspots serve as a referencing for the measurement. So that means that in one run you can run up to 36 interactions, and the main advantage of this instrument and this array system is that you can load your ligand into one channel, and in one run you can obtain up to six interactions for let's say for different concentration of your analyte, and from this you can calculate the association and dissociation constants. So that means what you can measure on ProteOn machine in few seconds, you have to measure for traditional approaches SPR for, let's say, several minutes or hours. So, and now I will switch to my scientific data. The first part of my presentation is interaction of Bordetella pertussis adenylate cyclase toxin with cell receptor CD11B CD18 integrin molecule. So on the slide there is a schematic representation how the toxin is interacting with the cells. Where the toxin is produced, the toxin consists of adenylate cyclase domain, which has adenylate cyclase activity. There are also some transmembrane helices and some big portion RTX domain, which is responsible for interaction with CD11B CD18 receptor. CD11B CD18 receptor is a receptor on monocytes, neutrophils, granulocytes, natural killer cells, and it is responsible for mainly for phagocytosis, and also it is important for cell-cell contact and some development of the embryo within the mother. So the toxin is secreted from the bacteria into the extracellular milieu, and it is recognized by the CD11B CD18 receptor, where they interact, and the toxin is able to embed it into the membrane and translocate directly from the outside of the cells into the cytosol, its adenylate cyclase domain, where has the catalytic activity, where the cytosolic ATP is converted to cyclic AMP, which produces the immunomodulatory properties, and in case of higher doses of the toxin, the death of the cells. The toxin also makes the oligomeric and cation selective pores, which also contributes to the cytotoxicity of the toxin. In case of CD11B CD18 receptor, this receptor mm, is belongs to the integrin family, which is a very big family of the proteins, which are the heterodimers. And you can see the schematic picture of the heterodimer, and these receptor can be found on the cells in several conformations. The one conformation which is seen on the left, this is the, let's say, band conformation, which is inactive, and low affinity conformations for the native ligands of the receptors, like complement C3. On the right, there is after activation of the receptor, there is a conformation switch into the extended conformation, which is active for native ligands, and this trigger this signal from outside to the cells, or also from inside to the cells to the outside. So, and I was interested which conformation of the toxin is recognized by this receptor. So I took the advantage that in our lab we established the expression of the recombinant CD11B receptor in the mammalian cells. So we express only ectodomain, so that means that this was expressed only the receptor without the transmembrane helix and the cytoplasmic part, and we were able to purify by immunoaffinity chromatography and by geofiltration. And by geofiltration, we were able to separate these two conformations. So in the upper part, there is a schematic. On the left, there is a schematic picture of the bent conformation, and the extended conformation is in the lower part. But if you analyze this fraction from the gel filtration chromatography on the negative on electron microscopy and the negative staining, we can see that we were able to separate the bent conformation from the extended conformation. And on the left side, there is negative staining, so which means that each white spots correspond to single ectodomain of this receptor. And at the bottom, there's not white spot, but some it's how to say, there is no white spot, but this correspond to the extended conformation of the integrin molecule. So by computational analysis and image analysis, we can observe by averaging, we can see nice band conformation of the ectodomain and the extended conformation of the ectodomain. So these two conformations was immobilized on the GLC ProteOn chip, and we can see on the next picture where the band conformation. This is a one-shot kinetics on the band conformation of the toxin with the band conformation on the left side, and the extended conformation of the molecule of the integrin molecule on the right side. And we can see slight differences in the sensorgrams. And I also did the control, that I used the MEM148 antibody, which is specific for extended conformation. And we can see that the binding of this antibody to extended conformation is higher in contrast to the band conformation. If you compare the binding of the MEM48 antibody, which is recognized only CD11B subunit, so we can see that the immobilization of the band conformation and the extended conformation is approximately the same. So that corresponds that the difference between the binding of the MEM148 antibody is probably, or is correspond to the changes in the affinity to the antibody to extended and band conformations. So by calculation, we were able to calculate the association and dissociation constant, and we can see that the toxin binds to the band conformation at 0.6 micromoles, while the binding of the toxin to the external conformation is about 2 micromole. So this means that there are different affinities of the toxin to extend it in the band conformation to the receptor. So on the next slide, there's another project which I did during my last year, and this is the interaction of analyte TRPV1 receptor with membrane-bound phosphatidylinositol 1,2-bisphosphate, BIP2, and TRP1 is a non-selective cation channel. It is polymodal receptor for various potential harmful signals, such as low pH or higher temperature or some specific chemicals, and it binds. It has 6 transmembrane helices, and both C and N termini are located in the cytosol of the cells, and these termini trigger the signaling of the receptor. And this receptor is able to bind the calmodulin, ATP, and BIP2, which modulate the function of the receptor. So we were interested in the N-terminal part of the receptor, which binds to BIP2. And first, we have to the setup of the instrument was the setup of the experiment was by immobilization of BIP2 enriched liposome to the chip surface. I used the chips with neutravidine, but recently the Bio-Rad launched a new chip, which is called LCP chip, and I recognize that for binding of liposome, the LCP chip is better than the NLC chip. So for the interaction, so this means that the liposome was immobilized to the chip, to the sensor surface chip, and this was done by paired oligonucleotides. So this means that in principle the one oligo is modified on the 5' by cholesterol, and by this cholesterol is embedded into the membrane of the liposome, and the paired oligonucleotide is modified on the 5' by biotin, and by this, so this means that we obtain the liposome, which is connected with double-strand DNA, and this DNA is able to bind to streptavidin molecule. So in this case, these liposomes were immobilized in these positions. So I tried two different approaches. One was the incubation of the liposomes with the oligos, and the second approach was the mixing of the lipid molecules with the 5' cholesterol oligonucleotide. So I recognize that the second choice is better, because if we incubate the paired oligonucleotides with liposomes, which are enriched for several types of lipids, so there should be some changes in the binding of the paired oligonucleotide to the chips. While if we use the binding of the 5' biotinylated oligonucleotide to already bound 5' cholesterol oligonucleotide to the liposome, the binding is is better because we obtain the specific binding of the liposome with different lipids, and the binding of these liposomes is quite, or is similar to the surface, is similar than in contrast to the mixed liposomes and oligo, because if we use some specific lipids like phosphatidyletanolamine or phosphatidylserine or some positively or negatively charged lipids, there is some interaction with DNA, and there will be some repulsion forces, and there is different immobilizations of the mixed liposomes with oligo. But if you directly incubate the 5' cholesterol oligo with liposomes, so there is in case if you use whatever lipids, the binding of the liposomes are quite the same or quite similar to the sensor surface. So on this slide, there's a kinetic binding of the C-terminal part of the receptor with liposome enriched with phosphatidylcholine and phosphatidylcholine with PIP2. On the left, there's a sensorgram of interaction of the C-terminal loop of the TRP1 with PIP2, while on the left there is a PC PIP2 liposomes, and on the right there is only phosphatidylcholine liposomes. So we can see that the protein is able to interact with the phosphatidylcholine alone, but the interaction with phosphatidylcholine, which is enriched with PIP2, is much higher in contrast to phosphatidylcholine alone. So we produce several mutants of the proteins, and we did the analysis, and we recognize that the wild type C-terminal part of the TRP1 receptor has affinity about 3 micromole, while if we did the specific mutation in the arginine 778, which is only a single mutation in this part, so that the affinity of the protein is decreased, let's say, in 2 orders of magnitude to more than 150 micromole. But if you change another lysines or arginines, like in the 770 or 781 or 785, there is no changes in the affinity of the protein. But if you change only 778, there is a change of the affinity. So from the data, I forgot the slide. So as I mentioned in the introduction for this project, so the C-terminal part of this receptor is able to interact with PIP2 and also with calmodulin. And we were interested if the binding of these 2 compounds, like PIP2 and calmodulin, could be shared by the binding site within the C-terminal distal region of TRP1 receptor. And as you can see on the upper part of the picture, there is an interaction over the surface enriched with PIP2 liposomes, and there is an interaction of TRP1 C-terminal part of the protein, and there is a nice interaction curve. But if you incubate the TRP1 protein with calmodulin and to produce the complex, so there is no binding of the complex anymore to the surface liposomes enriched with PIP2 molecules. So on in the lower part of the panel, you can see also the opposite evidence that there is no interaction of the PIP2 and camodulin, because if there is also surface which is enriched with liposomes with PIP2, there is interaction with TRP1 protein. The binding of the TRP1 protein is okay, but if you incubate this complex, the bound TRP1 to PIP2 on the surface chip with camodulin, there is no binding of camodulin to this surface. So from this data, we can conclude that both PIP2 and camodulin share the binding site within the C-terminal part of TRP1 receptor. So from this, we were able to make some model of the interaction of the C-terminal part of TRP1 with PIP2. So we produced the homology model, and we did the initial docking of PIP2 to this model. So we could recognize there are several sites for interaction of PIP2 molecule, but in the lower part of the panel, we can see that the PIP2 molecule is fundamentally bound to the specific helix in this protein, and this helix makes some triad of the arginines, the arginines 778, 781, and 785, and the side chains of the arginines are able to bind the PIP2 and the negative charges on the PIP2 molecules. And from this we can see that the arginine 778 is the most important residues, the most important residue for the binding of PIP2, for the recognition of PIP2 into these docking sites. So there's another project, and this is the third project which we did several weeks ago, and this is the interaction of a DNA binding domain of FOXO4 with target DNA sequence. So a few years ago, we obtained the crystal structure of this DNA binding domain of FOXO4 with target DNA sequence, and we recognize that there is the FOXO4 is a transcription factor that constitutes key component of conserved signaling pathway, and from the structural analysis of we were able to recognize that there are specific direct water DNA base contacts, and also there are unique water network interactions that contribute to FOXO binding to DNA in sequence-specific manner. So on the right part of the image on the slide, you can see the schematic representation of the binding of the protein to the DNA, and the W's, the circled W's, are specific waters which are in contact between the nucleotides in the DNA and the side chains in the proteins. So for this setup, so we immobilized the DNA oligonucleotides, the double-strand DNA oligonucleotides with specific sequence, which one is IRA, which is CyaA, and the second one was the DBE sequence, which is quite the same, but in the 5' there is only change from IRA is a CA, and on 5' of the DA is only GT, and the rest of the sequence is approximately the same. So for the setup, we used the LCP chip also, which is covered with neutramidine, and the oligonucleotide was adenylate and immobilized to the streptavidin sulfase chip. And we did the measurement of the wild type of the protein, of the foxo DNA binding domain, and we obtained a nice curve which were differing in the affinities between the ERE and the BA target binding sequence. And from calculation we can see that only the slight change between the CA and the GT nucleotides differ in the binding of the DNA, of the protein to the DNA, and we were able to calculate the association and dissociation constant, and we can see that the change between the CA and GT nucleotides between these two sequences is about the, let's say, 2 times or 3 times, 3 times higher for DBE than from the ERA sequence. So we were interested in the specific water contacts between these molecules. So we produced a lot of mutants. The one mutant is the tyrosine, the specific substitution of tyrosine to phenylalanine in position 102, and we can see that the association and dissociation, the affinity of the protein to DNA is slightly decreased, and this affinity, the decrease in the affinity is not due to association, but is due to dissociation. So if you compare these curves, the dissociation is about, this is not too slow in the wild type, but it's very rapid in this mutant. So this is another mutant which is the truncated mutant in position 184, and we can see that this mutant is not able to bind anymore. But as I mentioned, we produced a lot of mutants, and we produced 16 mutants in specific substitutions. And as you can see on the picture, there is a interaction kinetics between the 16 mutants, and between the 16 mutants and between the specific oligonucleotides like ERA or DBE. And this table, so this means that we did 16 mutants, and every mutant has two sides, so that we did 32 interactions and 32 values for the interactions. And as an example, that the ProteOn XPR is a big machine, is that this huge amount of the numbers we were able to obtain in few hours. So at the end of my presentation, I would like to conclude that the ProteOn XPR36 system enables rapid and high throughput analysis of various types of biomolecular interactions, including protein DNA, protein-protein, and protein and small molecules like lipids or steroids. And this instrument serves as a core facility at the Institute of Microbiology in Prague in the Czech Republic, and is intended for broad scientific research and some other interactions. So thank you for your attention.